A breakthrough that many people are waiting for in the medical field is the cure to cancer, and while treatments and therapies are improving every day, they are a tertiary response to this problem. One side of the medical field that is less focused on in the media is preventative treatments, which is why I wanted to concentrate on a project that focuses on preventing cancer. The biological problem I'm addressing is called Barrett's esophagus, which is a condition where the lining of the esophagus becomes irritated and damaged due to extensive acid reflux. This is usually a result of having gastroesophageal reflux disease, also known as GERD. In Barrett's esophagus, the squamous epithelial cells get replaced with columnar epithelial cells, which causes redness and swelling. This is usually diagnosed during endoscopies on the stomach or throat. Barrett's esophagus has also been linked to causing esophageal cancer, which is a somewhat rare but dangerous form of cancer. If this cancer cannot be operated on or treated with chemotherapy, one way that it can be treated is with a targeted treatment. One form of these treatments found to be successful for certain types of cancer focuses on the human epidermal growth factor receptor 2 protein, or HER2 protein. This protein is overexpressed in several types of cancer, especially breast cancer and esophageal cancer. The overexpression of this protein is linked with a higher mortality rate as well as a faster progression of the cancer, and this is mainly because the HER2 protein helps the cancer cells grow and spread. The HER2 is a receptor that interacts with other similar receptor and ligands to stimulate intracellular signal transduction pathways that are involved in cell growth. This protein has also been found to be overexpressed in Barrett's esophagus, so the targeted treatment I would like to focus on is trastuzumab, or Herceptin, which is a monoclonal or man-made antibody. There are 4 types of these man-made antibodies, and this one is humanized, which means that it's made from small parts of mouse proteins and attached to human proteins. This treatment is administered through an IV and can cause side effects similar to an allergic reactions like fever, chills, nausea, rashes, etc. This antibody binds to the HER2 protein in place of ligands that would normally activate the protein, and this activated protein would help the cancer cells spread and grow. Therefore, the antibody binding also significantly decreases the amount of cells undergoing S-phase and mitosis, which leads to less cell proliferation. This treatment has also been shown to prevent cleavage of the HER2 protein, so the protein receptor would not be able to leave the cell surface and travel to other cells where it might activate cell growth. The targeted therapy has been shown to work best when cells are overexpressing the HER2 protein, so this would work best on patients whose Barrett's esophagus also overexpress the protein. I believe that applying this treatment to Barrett's esophagus would be beneficial to patients because it could slow or even stop the spread of the damaged epithelial cells, which would reduce swelling and irritation caused by Barrett's esophagus, and most importantly, it could decrease the chance of Barrett's esophagus progressing into esophageal cancer. Overall, this would be an important development of treatment therapies because stopping the progression from Barrett's esophagus to esophageal cancer could save lives.